Today we're going to walk through the parasympathetic system, and this system is going to be a little bit easier to understand than the sympathetic nervous system, so that is a bonus. And then I'm also going to do some like quick YouTube video so that way you guys can see how to help your parasympathetic system, because as we talk about the sympathetic system, you know, the one for fight or flight, you'll see that this is where a lot of our stress issues come in, especially if it's chronic. So we do want to know, how do we help activate our parasympathetic system as well. So I put this in here because so many people often, I know as you're in school, you're like, I don't have time to take a break. I don't have time to relax. I've got like 1,000, bless you, I've got 1,000 things going on, and I get it because I've been there. But I really like this quote: the time to slow down is when you think you don't have time for it. Because if you are anything like some people out there and you burn the candle at both ends, whenever you don't slow down, you will be forced to slow down. Your body will make you slow down. So that's kind of the situation with my mom. She works 2 jobs and just constantly goes. I think she probably sleeps about 4 hours a night and does not eat healthy food. So whenever she crashes hard, and she's usually down for about a week. So whenever we're talking about the parasympathetic system, remember, life is about balance, and you do have to find times to work in that rest and relaxation, because your parasympathetic system has to also have some stimulation, not just your sympathetic where you're always go, 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 and do, and stress. So the functions of the parasympathetic division, this is also called the anabolic division. So make sure you know that that's anabolic, because what we're looking at is an increase in the nutrient content of the blood. Remember, this is the rest and digest system. So that digestion portion will allow us to help absorb some of those nutrients that our body needs in order to help repair and to build. So we're of course looking at relaxation for the rest portion, food processing, and nutrient absorption for the digestion. And remember, if we have misfiring of the parasympathetic nervous system, that can also lead to that faint and freeze, where we have the dominant parasympathetic nervous system causing inhibition to the sympathetic nervous system. So just don't forget that that portion is in there as well. So it's a little bit of misfiring, cross communication signals that can cause some of those issues whenever we're looking at that faint and freeze. So for our parasympathetic functions, this picture here will kind of show you some of the things that we're going to be looking at when we're talking about this system. Obviously stimulating the flow of saliva, because our saliva is rich in enzymes that will help break down the nutrients in our food. So we have salivary amylase, which will help break down carbohydrates. So the process of digestion actually begins in the mouth, and we need those salivary enzymes in order to help us with breaking apart our food. It will slow our heartbeat, of course, as we talked about a little bit the last time, constricts the bronchi, because we don't need to have dilated bronchi, we're not running a marathon. Stimulates peristalsis, which is the movement of the smooth muscle to help move the food through the GI tube. And then of course secretion, because we have lots of digestive enzymes that we will need as well. Stimulates the release of bile, which is important for emulsifying fats that you eat in a diet. So yesterday, whenever I was eating at Papados, the reason why my stomach is hurting today is because my body's not used to that, number one. And then number two, I'm really calling on my gallbladder to do a whole lot and breaking down all of that fried fatty food that I ate. So since my body isn't used to that, it's kind of having a little bit of turmoil today. And then contracting the bladder, of course, because whenever we're talking about the urination and defecation, we're going to be looking at that parasympathetic side. Okay, so these are just some of the things. If you prefer picture, you can use that slide that I just went through. If you prefer words, then you have basically those same things over here in the word slide. So whatever works best for you, it's perfectly fine. I think that the only one that wasn't mentioned on the picture was the changes in the blood flow and the glandular activity associated with the sexual arousal. So remember last time we talked about in order to kind of get into that state where you do want to have sex and get those glands flowing for the secretions, then yes, that is going to be a parasympathetic state. All right, so remember this is our craniosacral division. So today we'll be looking at how these things are going to come from the cranial portion as well as the sacral portion in order to innervate the various organs in the body. Remember, we have long preganglionic fibers, and then we're going to see the autonomic nuclei in the brainstem, and then S2 through S4. So that's your craniosacral, it's brainstem, and S2 through S4. We say that the ganglionic neurons are within or very close to the target organs. That's why there's the long preganglionic fibers, because they have a long distance to go in order to get to the ganglionic neurons within that ganglia, where they will synapse with the postganglionic. So therefore we have short postganglionic fibers that are in or near those target organs. So if we were to look at a picture of this, here's a good picture just to show you that long preganglionic neuron. Here is our ganglion, and then here is our postganglionic. So for your very long preganglionic, you can see here at the key, you'll be using acetylcholine as a neurotransmitter and acetylcholine as a neurotransmitter on the post as well. So it's very easy to remember the parasympathetic because they're both acetylcholine. Whenever we're looking at the parasympathetic preganglionic fibers that leave the brain, we're going to utilize cranial nerve 3, 7, 9, and 10 in order to get to various visceral structures within the head. Remember, this is craniosacral, so for the sacral portion, the S2 through S4 will form what we call pelvic nerves. So underline that, pelvic nerves. These are the nerves that are going to go on to innervate various structures within that abdominal pelvic cavity. Okay, so specifically closer to the pelvic region. So think about your bladder, the large intestine, the last portions of it, those sex organs. Those are all going to be in the pelvic cavity, and it will be under the influence of the pelvic nerves. If we look at this picture right here, 3, 7, 9, and 10 will use these ganglions in order to get to their target organs. So we're not just going to call it an autonomic ganglion, it actually has a name. And each one is named dependent upon which cranial nerve is going to flow through it. So you can see that cranial nerve 3 is going to go through the ciliary ganglion. Remember, cranial nerve 3 is oculomotor. So oculo meaning eye, motor being some sort of movement. And remember, for the autonomic nervous system, we're doing visceral effectors. So it's actual movement. So oculomotor is going to go to the eye muscles, which will change the pupil and the lens shape. Now, if we're in the parasympathetic system, which is for rest and digest, for these intrinsic eye muscles, are we going to dilate pupil or are we going to constrict pupil? Constrict, very good. Okay, so we're going to constrict. So just think about what you would do if you were going to go to sleep. For cranial nerve 7, you have the pterygopalatine and the submandibular ganglia that are going to go to these various organs here. So nasal glands, tear glands, salivary glands. And remember, it's all about the production of secretions. So we're going to stimulate these secretions for the parasympathetic side. And then the otic ganglion is going to go to the parotid salivary gland, again, to increase secretions, to help us with breaking down our food. And then we have cranial nerve 10 through the intramural ganglia. And this is going to be most of the organs within the abdominal cavity and the thoracic cavity. So like the heart, the lungs, cranial nerve 10 is going to be an extremely important one. So do not forget the vagus nerve actually will go inferior to the diaphragm. It is going to be the only cranial nerve that will go that far down into the body in order to provide that parasympathetic innervation. So very, very important cranial nerve. And then as we said earlier, the nuclei in S2 through S4 will form our pelvic nerves, also through the intramural ganglia to innervate the visceral organs in the inferior portion of that abdominal pelvic cavity. If you like charts better than pictures, then this is in here for you. So it basically kind of talks about some of the things that I already said in regards to what is the nerve, what is the ganglion called, where are we going, and ultimately what are we going to be doing. So you can use either one, whichever one makes you feel more comfortable and that you can learn easiest from, feel free to use them. They both have that same information. So remember I said cranial nerve 10, super important. 75%, you want to circle that, 75% of all parasympathetic outflow. So it's going to be vitally important that this nerve is healthy and that it does have the ability to help us with our homeostasis. Again, it is long. It's going to innervate structures beyond the neck and go into the two thoracic and abdominal pelvic cavities in order to get into various organs within that abdominal and pelvic region. And you will see that the branches intermingle with fibers of the sympathetic division. So that's kind of cool that they actually are running in those same areas. All parasympathetic neurons release acetylcholine, as we saw in our picture earlier. So again, like I said, kind of easy to remember that one because you don't have to remember some of the things that you'll see whenever we go through the sympathetic system. There's a lot of ifs, ands, or buts. So that one's pretty easy and straightforward. All neurons on parasympathetic side will release acetylcholine. Now, if we're using acetylcholine, any synapses that use that neurotransmitter are called cholinergic. Okay, acetylcholine will have a cholinergic synapse. So the effect on that postsynaptic cell is really going to be dependent upon the different types of receptors. And we have two types of cholinergic receptors. Do not forget these words. Muscarinic and nicotinic are words that you are definitely going to see in the test. So muscarinic and nicotinic are our receptor types. They're cholinergic. The effects of the parasympathetic stimulation of cholinergic receptors are localized and short-lived. So localized meaning, remember whenever we talked about the differences in the sympathetic and the parasympathetic, we said sympathetic can have mass activation because there's a greater amount of divergence. So this is more localized and it's not going to last as long as that sympathetic stimulation. Whenever we have the acetylcholine being released into the synapse, you guys learned this back whenever we were talking about the neurons, that we have to have this enzyme, acetylcholinesterase. So it's here again. Acetylcholinesterase is absolutely necessary because remember, we want to make sure that we inactivate the acetylcholine after it's been used, break it down, and then recycle and reuptake it so that we can continue to use it again. So most of that will be inactivated at the synapse by that enzyme, acetylcholinesterase. Any that diffuses into surrounding tissues is inactivated by tissue cholinesterase. So it just depends on if it's in the synapse or if it's in the tissue, but either way, we need to inactivate and we need to recycle and reuptake. So here's your quick reminder, kind of what we talked about a while back, in case you forgot. Choline and acetyl-CoA give us acetylcholine. This is the enzyme that will break it down into two portions. And then we're gonna reuptake that choline to use it all over again. So that process has to happen, or else we are not gonna be able to maintain homeostasis within our nervous system. Okay, so let's talk a little bit about these receptors. The nicotinic receptors, they are going to occur on the ganglion cells of both parasympathetic and sympathetic. Okay, so parasympathetic and sympathetic both have nicotinic receptors, as well as the somatic nervous system. So somatic nervous system, what was that one for? What did we innervate on somatic nervous system? Skeletal muscles. Very good. So remember, that's our voluntary portion. So the voluntary and the involuntary, ANS being the involuntary, all have these nicotinic receptors. Acetylcholine activates the nicotinic receptors, and it opens the chemically gated sodium channels in the postsynaptic membrane. And this is always, highlight, always excites. So always going to cause excitation of that cell. So let's go back to our action potential information for just a minute, because I know some of you got the sodium and the potassium a little bit confused whenever it came to action potentials on that particular portion of the test. So remember, we're at -70 millivolts as our resting membrane potential, right? It's when sodium comes into the cell, and sodium, is that positively charged or negatively charged?
Negative.
Which one is it?
Negative.
Not negative.
Positive.
Positive. Na plus. Okay? So since sodium is positively charged, you're bringing positively charged particles inside of a negatively charged cell. So are you going to make it more negative or less negative? More positive. Exactly. So we're going to go in the positive direction. When that positive sodium comes in, we're going to go in the positive direction. And remember, if we hit threshold, do you guys remember what the mark was for threshold?
-55.
Perfect, -55. So we have to make it more positive, and by sodium coming in, we're actually doing that. That will help give us our graded potential, and then if we hit -55, that gives us our action potential. So this is the excitation part of it, because the sodium is coming in and it's changing that measurement inside of the cell, which can then lead to the depolarization portion of the action potential, which takes us on the up end of the graph. You guys remember that, right? So down end of the graph was whenever potassium comes out of the cell, okay? So sodium comes in, that's the first thing. That will be excitatory to the cell. If we get the -55, we'll hit that action potential. Once we get to about that plus 30 millivolts, then we're going to have potassium come out of the cell. And because potassium is positively charged, when we move the potassium out, now our cell goes back down towards that negative state. Okay, so that was a recap, but just remember, the acetylcholine will open those gated sodium channels, and this is going to cause excitation of that cell. For muscarinic receptors, whenever we're looking at muscarinic receptors, these occur at what we call cholinergic neuromuscular, so a neuron next to a muscle. That could be a neuron next to a smooth muscle, a neuron next to a cardiac muscle, or neuroglandular. So maybe a neuron next to a sweat gland, or something like that. Neuroglandular in the parasympathetic division. Now remember, sweat glands are only innervated by sympathetic. So that was probably a bad example for me to use for you guys, okay? But a different gland. We have tons of glands inside of our body. So let's say the thyroid gland instead, okay? So neuroglandular, neuro, and a gland. These junctions here is where you will have those cholinergic muscarinic receptors. They also occur at the few cholinergic junctions in the sympathetic division. So now we're going to have these over on the sympathetic side as well. Okay, nicotinic and muscarinic are going to be in both sympathetic and parasympathetic. I know there's going to be a lot of icks. Muscarinic, nicotinic, sympathetic, parasympathetic. So try to keep it all separate as you are doing your studying and going through, because I know it can get a little bit confusing. Now, here's what's different. Highlight this. Muscarinic receptors are G protein coupled receptors. Okay, G protein coupled receptors are not going to work in the same way that I just talked to you about whenever we refreshed our information on how sodium comes into the cell and causes the excitation, which can lead to the action potential. So we're not going to be using sodium. We're instead going to be using the G protein coupled receptor, often abbreviated GPCRs. And these are a group of proteins that sit in the membrane. So here's your cellular membrane right here, your phospholipid bilayer. So you're going to have these proteins which will sit inside of the membrane, and they will bind signal molecules outside the cell. Okay, so first they got to bind. They got to bind it. Then they're going to transduct the signal into the cell. All right, so we still have something coming into the cell. And then they're going to cause that cellular response. Interesting point here is their stimulation produces longer lasting effects than does the stimulation of nicotinic receptors. So we have longer lasting effects because of the GPCRs. And this response can either be excitatory or inhibitory. It all depends upon the activation or inactivation of the specific enzymes. So there's a couple of different classes for muscarinic receptors. It's basically M1, 2, 3, 4, 5. I'm not going to go through all of the 5 with you guys, and I'm not going to make you memorize all of the 5 or learn all of the 5. You can remember the odd ones, 1, 3, 5, excite. Okay? The other 2, the 2 evens, are inhibitory. So whenever we watch this quick video, you're going to be able to see a 3D representation of what's happening with these GPCRs. So let's watch this really quickly.
Welcome to 2-Minute Neuroscience, where I simplistically explain neuroscience topics in 2 minutes or less. In this installment, I will discuss receptors and ligands. When neurotransmitter molecules are released from a neuron, they pass the signals to the next neuron by interacting with receptors on that neuron. These receptors are made up of proteins embedded in the membrane of the postsynaptic cell. There are 2 broad families of neurotransmitter receptors. One type are called ionotropic receptors. Ionotropic receptors have a site where a neurotransmitter can bind called the binding site. When a neurotransmitter binds here, it causes the channel to open in the receptor, and ions are permitted to flow into the neuron. This can change the membrane potential of the neuron and send a signal known as the action potential down that neuron. The neurotransmitter that binds is called the ligand, which is just a term for any substance that can bind to a target protein. Thus, ionotropic receptors are also called ligand-gated ion channels. The other type of receptors are metabotropic receptors, also known as G protein-coupled receptors. Neurotransmitters also bind to these receptors, but instead of immediately opening an ion channel, the next step after binding is the activation of an intermediate protein called a G protein. The G protein can then influence the opening of ion channels, but it can also affect enzymes and activate intracellular signaling molecules known as second messengers, which can initiate signaling cascades within the cell.
Okay, so a big portion that you already see different here is that the neurotransmitter, yes, it's still going to bind to the receptor, but we're not immediately opening an ion channel. Okay, so the ionotropic one is that sodium coming in, whereas this over here, the big key here is this G protein that sits here and is attached to this receptor. That's why it's called a G protein coupled receptor. So this portion is coupled to the receptor. Once the signal is sent, then what you can see here is there's going to be some enzyme activation, and there will be then changes in that ligand-gated channel. Okay, so that is the major difference between the 2.
G protein-coupled receptors thus tend to have a slower action, but can have more widespread effects due to their ability to influence various molecules throughout the cell. Ligands other than neurotransmitters, such as drugs, can also bind to receptors and have a variety of effects. If they have the same effect as the neurotransmitter, they are known as agonists. If they block the effects of the neurotransmitter, they are known as antagonists. Some drugs can bind to the site the neurotransmitter binds to and have the exact opposite effect of the neurotransmitter. These are called inverse agonists. Finally, drugs may bind to a site on the receptor that is separate from the site where the neurotransmitter binds and affect the likelihood that the neurotransmitter will bind. In this case, they're called neuromodulators and said to have an allosteric effect.
Okay, so why is he talking about drugs? Because in pharmacology, whenever drugs are produced, what they usually are targeting is G protein coupled receptors. That's how so many drugs actually do their mechanism of action. That's why they can have a widespread effect within the cell, because they're targeting that G protein coupled receptor, which can then affect multiple other things inside of the cell. Okay, we talk about that more in 2402, so I'll save that for later. So big forest picture for right now is whenever we're looking at nicotinic, we're talking about a receptor that is in the autonomic nervous system, both divisions, as well as the somatic nervous system. It's going to be using acetylcholine. It's going to be excitatory because we're going to have sodium coming into the cell because we are using a ligand-gated channel. When we're talking muscarinic, now we're talking about majority going to be on the parasympathetic division, only a few will be in the sympathetic side, and we're not using the same type of channel. Instead, we're going to use the G protein coupled receptor, and depending upon those enzymes that get activated or inactivated, we can be excitatory or inhibitory. M135 are excitatory, M24 are inhibitory. We have longer lasting effects because of all of the stimulation that can occur to the things that are inside of the cell, and this is what drugs are commonly going to target. Okay, you guys okay with that? All right. Let's see if we can go to the next slide. Okay, so as you're looking here, parasympathetic, because this is what we're talking about today, acetylcholine nicotinic receptors, we're going to have at the ganglia, postganglionic neurons also using acetylcholine, but using muscarinic receptors. So remember, this is where we have that neuromuscular or neuroglandular area for those muscarinic receptors. Now, this one uses what ion? Which ion did we say it uses?
Sodium.
Sodium. This one uses sodium or something else? Something else. What is this one going to use? G proteins. Perfect.
So I have a question.
Yeah.
I don't even know if it matters. So it looks like the G protein is already across that like cell layer, right? So is that?
You mean on the inside?
Yeah.
It's on the
That's
inside.
right. Okay.
Uh-huh. Yes. It is internal. And so whenever the receptor binds, then it signals for that G protein to become active. So it just sits there and just waits. Okay. All right. Oops. If we're talking dangerous environmental toxins like nicotine, this is where the name actually came from, this nicotinic receptor, because it can bind nicotine and it can be a very dangerous situation. So in regards to nicotine, the poisoning can occur if you have 50 milligrams of that compound ingested or absorbed through the skin. So when you're looking at tobacco, it's about 3 milligrams. So you can see that's quite a bit larger number than somebody who's smoking, although smoking is still very terrible and you should quit because it does so many bad things to your body. So the nicotine will bind to nicotinic receptors, and it will, if we have too much and it is a poisoning, give us these signs and symptoms. So SS is signs and symptoms. Vomiting, diarrhea, high blood pressure, you can see all of these things. Now, unfortunately, convulsions may occur because the neuromuscular junctions of the somatic nervous system, remember skeletal muscle, they are also stimulated. Nicotine poisoning can result in coma or death. So, got to be careful with that. Muscarine, this comes from the mushroom that is poisonous to our system. This will bind to muscarinic receptors, and it will give us signs and symptoms of what you see here: salivation, nausea, vomiting, diarrhea, constricting the respiratory passages. Now, obviously, we know that parasympathetic is going to do that, but it's going to do it to a whole different level to where you can't breathe. And then low blood pressure, so if your blood pressure drops too low, then you're going to have problems getting tissue perfusion, so oxygen and nutrients aren't going to be going into your tissues. And then an abnormally slow heart rate, which we call bradycardia. Anybody remember the term for abnormally high heart rate? Tachycardia, right, okay. So you can see here, basically, this is going to completely target the parasympathetic division because it's going to give us a much greater effect for all of the things that our parasympathetic nervous system is supposed to do, but to a very dangerous level. Whenever you get into nursing, you'll probably talk about atropine. So this is a drug that is considered anti-cholinergic or anti-muscarinic. It can block the receptors and be an antidote to these poisoning type situations, okay? So it will treat the poisoning because it'll block the receptors, help the diarrhea, the bronchospasm, the bradycardia, etc. I'm not going to go deep into atropine, but if you want more information on it, this YouTube video will talk to you more about the atropine. Okay? All right, so let's see where we're at thus far. Which cranial nerve is responsible for parasympathetic innervation of lungs, heart, GI tract? Vagus, perfect. Cranial nerve number what? 10, good. What neurotransmitter is released by the parasympathetic neurons? What'd you guys say? Acetylcholine, perfect. Acetylcholine is what we want. Synapses that use acetylcholine as a transmitter are called what? Cholinergic. Okay, so take that last portion out of the word acetylcholine, cholinergic. Okay. And then what are our two major types of receptors for the acetylcholine? Muscarinic and nicotinic. Very good. Think about the things that can poison you. Nicotine or those muscarinic mushrooms. So nicotinic, which blank causes excitation of that cell, and muscarinic, which can be what? So does nicotinic always cause, never cause? How would you fill in that blank? Always. Nicotinic, which always causes excitation of that cell, and muscarinic, which can be what? Excitatory or inhibitory, right? What is M1, 3, and 5? Is that excitatory or inhibitory? Excitatory, good. And then our even ones, 2 and 4, are inhibitory. There's your answers. Okay, so this is just a summary. As you go through and you think back, okay, what all have we talked about? We know parasympathetic is craniosacral. We know it's rest and digest. And we know it's anabolic. Those are all of the things that we've learned about the parasympathetic system so far. So rest and digest, craniosacral, and anabolic. Sympathetic nervous system, thoracolumbar division. We'll get into that more in the next class. So parasympathetic nerves, we know where they're going to leave the cord. If they're leaving at the brainstem, what are our 4 cranial nerve options? 3, 7, 10, and what else?
8, 9.
9. Okay. All right. Sympathetic pathways, we'll talk about those next time. The fibers leaving the brainstem or the spinal cord terminate in autonomic ganglia. Remember, we have different names for our autonomic ganglia in the parasympathetic system, depending upon which cranial nerve will be going through there. And then we have the preganglionic fibers and postganglionic fibers. Remember, we're releasing acetylcholine. For preganglionic, we're releasing acetylcholine onto nicotinic. For postganglionic, we're releasing acetylcholine onto muscarinic. And we'll do all of the sympathetic stuff the next time. Okay? So, whenever we're talking about our parasympathetic system, rest and digest, of course, digestion is a huge portion of this. we've already learned that for our digestive system, we have the enteric nervous system, which does have the ability to work outside of the control of the CNS. So remember last time we talked about the myenteric plexus and the submucosal plexus. So this is where we have our ENS neurons in those two plexuses to help us with our homeostasis in our digestive system. So whenever you're looking at the intimate contact with foods, liquids, toxins, chemicals, anything that can come into your body, it's for this reason that we will have ENS communicating efficiently in order to help us with maintaining homeostasis beyond just the gut. So remember, this goes into mind, mood, circulation, even energy levels, because if you're not eating good, healthy things, you're not going to feel energetic because your cells are not able to get the nutrition that they need. So the enteric nervous system's role in regulating our overall physiology and even psychology should make it very clear to you, if I haven't already beat this like a dead horse this whole semester, that what you put in your mouth is incredibly important, but even so, more than how you choose to exercise. So now here we're introducing a new topic. Yes, exercise is important. There isn't anything that you can do to replace it. There isn't a supplement, a pill, a laser, nothing that you can do to replace exercise. So incredibly important, but there's a lot of people who eat like crap and think that they're gonna go work it off at the gym. And that's just not how it works. That's not how our system is set up. So whenever you consider each meal influences your physiology between 55 to 72 hours under normal conditions, and you may only work out for a few hours each week, obviously the food has a much greater capacity to alter all three sides of the triangle than exercise done only a few times per week. So if that's the case, yes, we need to eat good, healthy foods. We need to nourish our bodies. We need to make sure that we have all of the nutrients that are necessary in order to help us with repairing and rebuilding. But what else can we do? So other than just eating good, because we even sometimes have our days when we're like, I'm not going to eat good. So this quick video will kind of give you an idea of how else we can help with regulating our parasympathetic nervous system to help control the different parts of the triangle.
In this episode, we're going to talk about your vagal tone.
Vagal tone, cranial nerve what? 10.
So in a previous episode, we talked about 4 self-regulatory techniques that help trigger the parasympathetic response in our nervous system. That's the calming reaction that our nervous system has to balance out the sympathetic response, which is the alerting fight, flight, or freeze response. In this episode, we're going to talk about a few more things that we can do to calm our brains. One of the first things that comes to mind is meditative breathing. So long, deep, intentional breathing. Now, this helps trigger that calming reaction, and people have been doing it for thousands of years for that very reason. When you breathe out with a long exhale, you may notice an increased production of saliva in your mouth. Now, that's a parasympathetic response. A lot more people are familiar with the sympathetic response, which is a dry mouth. So, meditative breathing, long, slow out breaths. Now, on that note, mindfulness and meditation is also very helpful in triggering the parasympathetic nervous system, and we'll talk more about that in upcoming episodes. Laughter is another technique that helps trigger the parasympathetic response, so try and find a way to laugh every day, even if it's watching dumb cat videos on YouTube. I'm going to put a link to my favorite down in the description, so check that out. Another way to foster your vagal tone, which is the strength of your vagus nerve, is to listen to your biorhythms. So that means going to sleep when you're tired, waking up when you're refreshed, eating when you're hungry, stopping when you're full. These sort of biorhythms naturally help the body restore its nervous system and function at its optimal level. We all know how crappy we feel when we don't have enough sleep and how it affects our emotional reactions. Now, multitasking is something our brains are not very well developed to do. So monotasking is a way that helps calm the brain, doing one task at a time and doing it slowly. So doing one slow thing a day, something that's calming and slow and peaceful. For example, sitting down and petting a dog, or taking a slow walk, looking at the sunset or doing some knitting or crocheting, whatever it is that you like to do, but just do it slowly without rush. That helps trigger that vagal response. Social connection also helps trigger that parasympathetic response. So hugging someone, interacting in a positive way, reaching out and connecting, that all helps soothe the mind and foster calmness. Anything we can do to take care of our body also helps relieve that sympathetic, stressed out, stored up, pent up energy. So stretching, like just stretching out your muscles, is a helpful way to release that tension and trigger that calming reaction, as well as getting a massage or any kind of physical activity really can be very helpful. Another weird thing that triggers the parasympathetic response is standing on your head. Now, this puts extra pressure on the heart, which causes the heart to react differently. And long story short, it triggers that vagal nerve messaging to the body to slow down and calm down. So you can give that a try. Go try standing on your head. Now, it's important to remember that none of these techniques are going to solve your problems. The purpose behind these techniques is calming you down so that you can solve your problems. If we use these anxiety-reducing techniques as a distraction from our problems or from our anxiety, in the long run, our anxiety is going to stay. So it's really important to find out the function behind your emotions, what they're trying to tell you, and then meet their needs. And we'll talk more about this in upcoming videos. But make sure you're not using these techniques as an attempt to control or force your emotions to change, as that can often make things worse. What we're really trying to do is foster the calm part of your nervous system that's already there. Hope this was helpful. Thanks for watching and take care.
Okay, so I'm going to add one more thing to it that she left off, and this one is actually really important for people who are stressed, but they don't know they're stressed. All right, so this was definitely my case, because I heard tons of people as I was going through my doctorate program, you need to like do something to relieve your stress. And I'm like, I'm not stressed, I'm fine, I'm not stressed. So I did not know how to identify stress. For those people who you hear that constantly and you're like, I don't know why this person is saying this, I don't know what to do in order to help myself, like I don't even know. The answer to that is acupuncture, okay? Because acupuncture will let you feel like what you're supposed to feel internally whenever you're not stressed out. So whenever I first went and got acupuncture, I did not know that this was going to actually occur. It was one of my friends that I used to work with at a clinic years ago. She told me that she had somebody that was trying to practice acupuncture, and could I basically be a guinea pig for him as he worked on perfecting his technique. And so of course I was like, yes, because as I was a student, you know, I needed people to allow me to work on them as well. So I always try to give back to all of the students. That's why you'll find me in like, you know, the cosmetology schools getting my hair cut and like just praying, oh please God, let it grow back fast. Okay, but I always give back. I go to the dental hygiene center at Collin for students. Like I do try to do it all. So this guy came to our office one day and he was like, okay, well just anything in particular. And then my friend was like, just do the stress protocol for her. I'm like, I really don't have that much stress, so I don't even know what she's talking about. So he did his little protocol. I lay there for 15 minutes. He took all of the needles out. It does not hurt if you're scared of needles or anything like that. You can close your eyes, you won't even feel it. Does not hurt. So I got up and I was like, I'm like walking slowly around the office because I'm trying to figure out what's happening. And I'm like, I just feel like I couldn't explain it. And I was trying to tell her, and she was like, you mean you feel like light, like you feel better, like is it good? And I was like, yeah, it's like real good. Like I don't know. It's kind of like a feather, like a cloud. I don't really know. And she looked at me and she was like, I'm gonna tell you exactly what she told me, okay? Bitch, that is what you should feel like. That is normal. You don't know what normal was because you lived your whole life stressed out, like the clap and everything. I was like, you know what? This is amazing. I was like, this is how people walk around feeling? And she was like, yeah, that's how people walk around feeling. So it was an eye-opening experience for me because I never knew what to do. And it seemed like even if I followed all of these techniques that everybody talks about, it wouldn't make it better. Massage didn't help. I would still feel achy afterwards. So all kinds of things I tried, but acupuncture was really that key for me, just to kind of help rebalance what's known as the chi, or the energy inside of the body. So if you've never tried it before, I highly encourage you to try it, especially if you are stressed out, okay? All right, so I think that is going to be the end. Oh, last slide. Every day may not be good, but there is something good in every day. Sometimes it's just telling ourselves that positive aspect of things, or finding hope in something that can also help draw us back to our parasympathetic nervous system and not allow us to be so stressed out. So that is what I got for you for the parasympathetic side. Next time we are going to do the sympathetic side. Anybody